Hello and welcome to week 8. This week we're going to focus on chapter 13. In this chapter we'll talk more about the genetic code and transcription. So to begin with, we really need to talk about the central dogma of molecular biology, and that is that DNA makes RNA and RNA makes protein. This week we're going to talk about the first half of this, or how is RNA made? What we know is that genes code for the sequence of nucleotides in RNA molecules. And then during translation, this RNA can bring about formation of a protein coded for by DNA of that particular gene. But this week we're really going to focus just on the first half of this. And so one way to think about this, at least in terms of humans, is that DNA is our cookbook. It's a cookbook to make you. In this cookbook, we have 23 chapters, or 23 pairs of chromosomes. Every gene that we have could be considered to be a recipe. And in this, RNA not only is the cook, but it's also a copy of that recipe that the cook might be carrying around, like on a little index card. Okay? The end product of this, the end goal of this DNA, is to make various proteins. So during transcription and translation, what happens is that you've got your DNA template. Okay? This DNA is going to contain your genes, and it's going to serve as a template for this RNA formation. Right here we've got our DNA template strand, and through transcription we're going to make a copy of this. This copy is going to be complementary to our initial starting strand. We're going to go through this a little bit more next week, but some of the homework questions focus on this from Mastering Genetics, so let's go through this. So what happens here, we have some sequence. Okay, we've got TAC, CAC, AAC, and TCG, and this is what's found in the template strand. After transcription, the mRNA is going to be complementary to that DNA. The only difference is that anytime you would normally want to write a T that would be complementary, you can't, because RNA does not have thymines. So instead you have to write a U for uracil. So in this case, a T would bind with an A, and that's indeed what we see. An A would normally bind with a T, but we can't have a T again, so it has to be a U. A C with a G, a C with a G, A again has to be a U, no thymine, C with a G, and so on. And so these would be complementary to one another. We're going to talk more in a minute about these, what we're going to call codons. Codons are going to be groups of 3 nucleotides that are read together. Later on, when we talk about chapter 14, we'll talk about translation. What happens during translation is that you have an mRNA transcript that is going to be directing the sequence of amino acids in your polypeptide. That translation will happen at ribosomes. So let's talk more about this triplet nature of the code, or what a codon is. So to begin with, in the 1960s, Sidney Brenner argued that this code must be a triplet. And the reason for this is because if you look at the math, we've got 4 letters, or 4 bases that you can use. And you need to specify 20 amino acids. Okay? So if you only had 2 letters, okay, that would give you 16 possible combinations, and that's not enough to cover that 20 essential amino acids that we need for our proteins. If you have a triplet, if you have length of 3, that would give you 64 possible combinations, and that's indeed what we see. That is more than enough to specify our 20 amino acids. He also argued that longer codes, such as a length of 4, would just be too cumbersome, and in general everything tries to be as efficient as possible. So having all of these extra ways to specify just those 20 amino acids, probably not likely to happen. And so what they did, there was a number of different scientists, Crick, Barnett, Brenner, Watts-Tobin. They performed a number of insertion and deletion mutations. And what they found was that this reading frame of 3 bases, of 3 letters, was absolutely critical. What happens, or when we talk about these frame shift mutations, is an insertion or deletion of 1 or possibly more nucleotides from the DNA. And I think the best way to illustrate this is to use actual words rather than just the bases. So if we made up a sentence, the normal sentence might read something like, the cat ate the rat. If you're going to delete this C, everything downstream is going to be shifted over. Remember, they have to be read in groups of 3. So if everything is shifted over, what might happen? You've still got the, that's fine, but that C is missing now. It'd be at a, tet, hair. I mean, that doesn't make any sense, right? After insertion, the same thing, okay? You add an extra C here. Everything downstream is shifted and out of frame, and now this doesn't make sense. So that initial reading frame, that frame of 3 codons, absolutely critical for an organism. If you get these insertions, if you get these deletions, and such that everything is read out of frame, everything downstream from that insertion or deletion is basically going to be nonsense.